Bismillahirrahmanirrahim. Today I'm going to present in the third international conference on applied zoology, a virtual conference. I'm very thankful to the organizer for giving us the opportunity to present here in this forum. So the topic of my research is Bellis perennisa, common ornamental plant with a potency of lowering glycemia and associated health biomarkers. I am Dr. Haseeb Anwar from the Department of Physiology, Government College, University, Faisalabad. So the outline of my presentation is about the pathogenesis of diabetes and the role of oxidative free radicals in the onset of diabetes, its aims and objectives, and the results which we obtained from the study. So about the pathogenesis of diabetes, there are two types of the pathogenesis. One is the genetic predisposition and the environmental factors. Collectively, both ultimately cause the decreased glucose uptake in the muscles, and it will lead to the hyperglycemia, and ultimately it will lead to the type 2 diabetes, which is actually due to the resistance of the insulin in the receptors at the targeted cells. If we talk about the role of the oxidative stress in the diabetes, due to the hyperglycemia, glycolysis cycle is disturbed, the mitochondrial metabolism is disturbed, the generation of free radicals, and the activation of few pathways involved in the stress signaling. Ultimately, the free radicals, they will cause DNA damage, lipid peroxidation, protein damage, and the stress signaling. Collectively, these will cause three major things. One is the insulin resistance, which leads to the type 2 diabetes mellitus, beta cell dysfunction, which leads to the diabetes type 1, and ultimately both of these types could lead to the diabetic complications. So this is a common daisy plant, family Asteraceae, and it has phenolic compounds mainly, and it has different beneficial health effects like anxiety, memory learning, and mainly it has anti-hyperlipidemic antioxidant effects, due to which we have selected this plant to test it in the management of diabetes mellitus. The objective is to evaluate the anti-hyperglycemic, antioxidant potential, immunomodulatory, hepato-protective effects of this plant flowers ethanolic extract in the diabetic rats. If we talk about the experimental model, we have 5 groups comprising of 8 rats in each. One is the negative control without any treatment, without induction of diabetes. One is induced diabetes but without any treatment, and 3 groups comprised of the treatment. One of them is receiving the standard control drug, and the rest of the 2 groups will receive the plant ethanolic extract at 2 doses, 100 milligram and 200 milligram. We induce the diabetes in the rats by using the streptozotocin and nicotinamide injection, and the blood glucose level above the 200 milligrams per deciliter was considered as diabetic. So 3 days were taken for that, and we started the supplementation from the day 0, and the total experiment is comprised of 21 days. And at the end of the experiment, at 21st day, we actually decapitate the rats, and we collect the serum samples for the further analysis. These are the different analyses which we done. The anti-diabetic activity was comprised of 3 hormones: insulin, glucagon, and the glucose. For LFTs, different enzymes of the liver and albumin and total protein was counted. And to assess the oxidative stress, we did the total antioxidant capacity, total oxidant status, peroxidase, and aryl esterase activity in the serum. For the immunomodulatory activity, we did the DNCB assay. About the results, it was seen that the glucagon hormone and the glucose level is significantly decreased in all the treated groups, including the standard control drug group and the ethanolic extract group in the glucagon and in the glucose results. However, in the insulin, the results were very significantly interesting, as the insulin level was significantly increased in the treated groups, particularly the standard control group and the 200 milligram ethanolic extract group, which was a good sign. And if we talk about the LFTs, the ALT and AST was significantly reduced in the ethanolic extract treated groups. However, the alkaline phosphatase is also significantly reduced. That reduction was in compared to the positive control and the standard drug control group. Albumin was also significantly decreased in all the groups as compared to the negative control, and the total protein, it was increased in the standard control group and the positive control group. If we talk about the oxidative stress, the It was very interesting. It is comprised of very interesting results. The total oxidant status was significantly reduced in all the treated groups as compared to the positive control. And see, similarly, total antioxidant capacity, it was significantly increased in the treated groups as compared to the positive control. However, the peroxidase and aryl esterase enzyme activity is also similar to the total antioxidant capacity activity. It was significantly increased in all the treated groups, which shows the good antioxidant effect of this plant ethanolic extract. If we talk about the immune modulatory activity was significantly increased in the 200 milligram ethanolic extract group at day 3 after this DNCB. So if we see the overall results, we can say that this plant extracts due to its phenolic contents, it reduces the oxidative stress, and ultimately it plays an antioxidative role, which helps to correct the hyperglycemia. It improves the beta cell function, it enhances the liver functions, and it also boosts the immune response of the body. Collectively, all of these effects were actually helpful, and you can say the responsible to reduce the oxidative stress, which ultimately in turn reduces the hyperglycemia by improving the beta cell function, and it also boosts the immune response and ultimately the liver function as well, which is a very good sign. And ethanolic extract, conclusively we can say that the ethanolic extract of this plant, daisy plant, plays a significant role in managing the diabetes mellitus by reducing the oxidative stress, and it also shows the hepato-protective effect and the immune boosting effect in the hyperglycemic conditions, which was artificially induced in the rats. So thank you very much for your kind intention. I think this presentation will be very interesting and food for thought for all of you people. Thank you very much for kindly listening. I'm thankful.